And thank you everyone for joining us today. My name is Nadia, and with me today, Mubariz, Aniqa, Kunwar, and Maral are here as Health Sci 403 students to host this Demystifying Medicine webinar series. Before I introduce today's speaker, I would like to ask Aniqa to do a land acknowledgement.
Thank you, Nadia. So we would like to begin by acknowledging that McMaster University recognizes that it is located on the traditional territories of the Mississauga and Haudenosaunee nations and within the lands protected by the Dish with One Spoon Wampum Agreement. This agreement is also known as One Dish, One Spoon, and it is a law used by the indigenous peoples of the Americas since at least 1142. And essentially, it's an agreement for sharing hunting territory among two or more nations. So with that acknowledgement, I will pass it back to Nadia for an introduction to our amazing speaker today.
Thank you, Aniqa. So today we are fortunate to have Professor Simon Rousseau as an associate professor in the Department of Medicine at McGill University. He's a researcher in the translational program in respiratory medicine at the Research Institute of the McGill University Health Center and the McGill Whole Person Care programs. Professor Rousseau is also the director of the Quebec Respiratory Health Network Biobank and has served the needs of the scientific community to perform high-quality translational research. Since 2018, he is the director of the Quebec Respiratory Health Network Biobank, a province-wide infrastructure that coordinates the collection, storage, and distribution of human biosamples relevant to lung health to researchers locally and worldwide. His research focuses on the intracellular signaling mechanisms regulating inflammation to understand the role of chronic infections in neutrophilic inflammation in cystic fibrosis. Today, he will be talking about the importance of Biobank, especially during the pandemic. So please welcome Professor Simon Rousseau.
Thank you everyone. Thank you, Nadia, for the kind introduction. I'm really happy to be here for this demystifying seminar, which will be really focusing on Biobank. And for some of you, Biobank may be kind of intuitive in what it does. It banks biospecimens, but underneath there's a lot of steps that even most researchers may not be aware of. So I'm going to take time today to use the project we had in setting up a biobank for the COVID-19 pandemic to illustrate some of those important steps in creating the biobank. Although I'm a basic researcher, I was trained in cell and molecular biology. I got to be involved in biobanking, which sometimes may look a little bit as a surprising detour. Although right from the start, when I was doing my master's degree, we were collecting cells from umbilical cords to do our work. So maybe I was predisposed to work with human-derived biosamples. Let me just move to the next slide. Okay. Since as was mentioned, in 2018, I direct the Quebec Respiratory Health Network Biobank. So it's a network of different institutions who decided that sharing their resources would be really helpful in helping other researchers perform research on those precious human biosamples. And in that role, and leveraging the expertise we had, I was invited to participate in the Quebec COVID-19 Biobank that was set up in March 2020. So the objectives for today is really to describe what role biobanks can play as key infrastructure in research, more specifically towards COVID-19. Always in this context, we'll look a little bit at some of the opportunities and challenges that were associated with setting up the biobank under these conditions. And I'll finish up very briefly showing what can be done, or what at least we're doing with some of this data, using what's been called a post-theory science approach. And I'll tell you a little bit what I mean by post-theory science, and see how Biobank can enable that type of research. So the focus of the biobank will be on samples that were collected in Quebec. So I'll present a few numbers, but obviously the COVID pandemic affected worldwide and throughout Canada. But because of the nature of the samples, I'll give you some of the number within the provinces here. And so far, these numbers were taken as of this weekend. We're getting close to 1 million confirmed cases. So obviously, there's a lot more cases than those that were confirmed in Quebec. Since January, a lot less testing has been done, formal testing. And on top of that, there's all those cases that wouldn't have been numbered. Number of deaths were at 14,245. So this is far from being insignificant, and therefore highlights the need of better understanding COVID-19, but any other health problem that may emerge, such as the current pandemic that we've seen. And when you're tackling something new, you have to engage the research community with in different ways to try to understand what's going on. So early on in the pandemic, the provincial government decided to act in different ways, but one of them was to set up a biobank. And there was a mandate that was given to achieve this, which was to support the research and identify clinical and molecular determinants of COVID-19, susceptibility, severity, and outcome. So to do this, the FRQS, which was behind this kind of the early driver of this initiative with Genome Québec, they've assembled a group of researchers so that we could use our prior expertise in setting up a biobank. So the biobank itself, I'll give you its structure right away, but we'll go down in a lot more details as we move on. So the biobank is set up with a steering committee at its core. So these are researchers that meet together. We're still meeting every week. We were meeting every day early in the pandemic to set up the biobank. And we're really responsible for managing the operations of the biobank. The steering committee itself is overseen by a governing committee that is composed of stakeholders represented mostly by directors of the different research institutes in Quebec that host the researchers that are interested in using the biobank data. We have the opportunity, and we're lucky to have an international scientific board composed of members from the United States, UK, Switzerland, which help us determine what are the key questions, key scientific questions that are important in general, so from a worldwide perspective, and also to connect us with different initiatives so that we can really network our samples and data with researchers throughout the planet, and also find other collection to which can be really helpful. There's also a access committee, and I'll come back to this. It's an independent access committee that is responsible for evaluating applications for access to data and sample. And then the steering committee is broken down in different subgroups that have different tasks. Some are linked with communications, operation, mostly budgeting. There's ethics, a very important component. Data management as well. This is becoming huge. As you'll see, we've gathered important sources of data, and there's different subgroups that are illustrated here. So you can see that it's not a super heavy structure, but it still needs collaboration with the different groups of people that have all decided to dedicate time to do this. So now, what is a biobank? Why would we want to create a biobank to help research on a disease like COVID-19? So there's a biobank in essence. I think the name says a lot about it. It's a repository of biosamples that are linked to clinical data. So that's really important. A good biobank will have a lot of clinical variables that are linked to the biological material, so that you can actually study the outcomes that are of the biology, or the fundamental molecular biology that you can discover through the biosamples, and the clinical outcomes that are associated with those samples. So that link is really a core component of the biobank. It supports all kinds of research, but I've included here personalized medicine because this is one of the well, it's an area that we've been hearing more and more about, but which biobanks are really well placed to answer, because we can still have multiple participants reflecting different groups in the population, and therefore understand the differences in terms of disease progression, response to treatments, and personalized treatment based on those subpopulations. In as much that we can recruit sufficient participant that reflects the diversity of people affected by certain conditions. And so that leads kind of to the second point. The strengths of a biobank is really the capacity to gather several participants so that we can enable large studies. Cohort studies are really well suited for that. And biobank by themselves can be, they can collect from different studies. So they're not necessarily linked to only one study. What we'll be looking today is a special case for COVID-19 where the cohort study is embedded in the biobank itself. If I look at the Quebec Respiratory Health Network biobank, however, there's dozens of studies currently active that are collecting for their own purpose, and the excess material is being put in the biobank for usage by other researchers. So there's more than one model of biobanking, but really it's collecting these samples together, and that allows a lot of researchers to access them. And what's really neat as well is the breadth of questions you can now ask. You're kind of maximizing what can be done with the samples and the data that's been collected, because it's not only the initial question that can be answered, but the resource being opened up to the research community more at large. Then there's kind of a possibility of looking at the data from different angles, answering a lot more questions, and that brings more researchers in exploiting this really rich resources, and also increases the breadth of questions and the depth of the science that can be carried out. That's, I think, two very important points about the role of biobanks. Now, we'll look at the BQC19, which stands for Biobanque québécoise de la COVID-19, which is the Quebec COVID-19 biobank that was set up. Give you just a brief overview of how it is. So as I said, the biobank itself, it's kind of the repository, this area where you get the data the material and data that's been collected, and where the eventually researchers can apply for access and get access to this data. So it starts, I probably should click on the cursor so you can see it better. So in this case, initially we had 11 hospitals participating. Now 2 are not actively recruiting anymore, so there's still currently 9 hospitals throughout the province. The majority of them are situated in Montreal, but not only in Montreal, but we cover 5 different areas where participants are recruited based on having been tested using a PCR test for SARS-CoV-2, so for the virus that is responsible for COVID-19. And whether the test is positive or negative, that's the sole inclusion criteria, so that enables us to have control. So participants that have tested negative, but still can be included in the study to increase the power of making discovery when you want to compare COVID-19 positive versus COVID-19 negative, for example. When people are recruited into the biobank, we will collect clinical data, blood samples, and they are stored. Something that is particular to the COVID-19 biobank is the fact that we're actually doing some analysis that we're providing for the entire community, and I'll come back to this. So we are generating some experimental data that is to be exploited by everyone that applies for access. And that's been, I think it's a very interesting and neat way of exploiting these resources using the funding that was provided by the different agencies. And I think it is visionary from our funding agencies to have seen the utility of generating experimental data that would be available to all researchers. And as I said, I'll give you more detail. The last part that is obviously you have to provide access. This information that is being collected, including the clinical data, will be denominalized, so we remove any identifying identification, but there are strict ethics rules to which I will be coming, and that means we have to control access. Any research group that want to use the biomaterial and or the clinical data or experimental data that is found within the biobank have to have ethics approval from their own institution in order to have access. And so we have a committee that looks at these questions in addition to other ones. So we'll now go into a little bit more details. And while I'm here, I'd like to acknowledge a colleague in BQC19 and collaborator on different research projects, Karim, who graciously provided me with a few slides that were neatly designed, such as this one, making the design of the biobank clearer. So if we go on to, again, the last part of a biobank, which is, I kind of alluded to it, is the ethics and the policies, which are actually crucial elements to a biobank. And to be fair, I wasn't trained in policies and documentation and ethics as a researcher, doing mostly simulation of cells. So this is an aspect that sometimes we may overlook, but it is truly important. Because of that, I'll take you through the next few slides to illustrate what some of these key documents are and what are their purposes. Because, you know, when you access a sample, you don't always realize all the work that's behind getting those samples, but not just from the clinical side, but just from the policies and regulatory side, just to make sure that that everything is done ethically, that it is done within a legal framework, and also that the procedures are normalized. So that demands a lot of documentation that we need to generate. And that's even more, it illustrates some of the challenges that you may encounter when you're trying to set up a biobank quickly. So the other, I guess the flip side of this is it illustrates what needs to be done when if you're thinking of setting up a biobank. There's a lot of groundwork to be done ahead of already starting the collection and distributing the material. So the core document that would be describing most of what's going on in the biobank is called the management framework. It's the document that details the governance, the policies of the biobank. It's really the biobank on paper. It will say where is what, who's doing what, consent procedures, how do you store the material. So a lot of this information is collected in the management framework. It's a document that you have to submit to ethics and with the in the consent forms, and it will change depend as the time goes by. And to give you an idea, in the case of the Quebec COVID-19 Biobank, you see here this is the first page of our management framework, and maybe it's kind of tiny. This was the version 11, so there was already 10 prior version. That's the one that was approved in October. We're already at version 12 right now, where we add details if someone changes their function. Sometimes the recruitment criteria may vary a little bit. Any procedures that is changed has to be updated into the management framework. You can also see it's a 41-page document, so it's not such an easy thing to see. Now you'll see on the screen, and I'll try to make it bigger on mine, the different headings of that document. So it covers everything going from the objectives, and the BQC19 framework is available to all on our website, but it is in French, so I'll do a quick translation of those headings to give you an idea of its actual content. So you have the objectives. We describe where are situated the physical aspect of the biobank, the persons that are responsible. You detail the financing of the infrastructure. All recruitment procedures are very well detailed within the biobank. You details the biological material that it's going to be collected, the data that's being collected. So what are you collecting, how it's going to be stored, In this particular management framework, we also have core analysis that I'll come back. So these are detailed here. We talk about the policies for publication and access to the data and material. The governance is described. The diagram I've shown you before is also illustrated here. You'll excuse me, the file on my computer is quite small, so that's why I'm struggling a little bit in reading some of those headings. The access committee, this is the committee and the procedures that describe who can access the samples, what are the criteria. So this is an important section as well. And there's a lot of annexes that details all of this. So this is an important document that details everything that are going on in the biobank. In addition to that, there are other key documents. There are consent forms. So these are the forms that will describe that the patient will sign, that describes what are the responsibilities of the biobanks towards them, what will be the purpose of the research for which they've just consented. So this is a key document. In the case of the COVID-19 biobank, and I'll show you the consent procedures in a couple of slides, but we also had to have not only a written form, but verbal script because of the containment measures. So I'll give you a little bit more details on this. But you also want to provide detailed manuals of operations. So these are documents that tells the teams at each site what they have to do, what are the criteria for recruitment, what are the procedures for consent, what are the procedures for collecting blood, what you write on the bags, where must the blood go, and then once you take the samples from the bedside, they have to be taken to the lab for processing. And it's important that the processing is done the same way from one site to the other. So again, all blood processing, you prepare your protocols, and these are very detailed protocol that you send to different sites. So these are the types of document that you'd find that are submitted to each site that are participating, and then they just follow these diagrams that's telling them once you encounter a patient, this is what you should do, whether they can be recruited or not. This is the script that you must read to them for consent. This is the number of tubes that should be taken for the blood collection. These types of tubes go for example, plasma preparation. Other types of tubes, such as PAX tubes, may be used for nucleic acid extraction, and so on and so forth. Again, these are big documents, crucial in ensuring that you have a high quality biosample set and data set, because this is what sets the conditions. And the strength of a biobank, obviously, is that everything is done the same. It's like everything else in science. If you want to compare things, you want to minimize variations. And for that, you standardize your procedures, so these famous SOPs, and these manuals that you send to the different teams, and to ensure that they follow them correctly. We also have a very detailed case report form. So that's another set of document that has to be prepared in advance. That's basically all the fields you want to collect in terms of clinical information so that you can put this in the database. And we've used a data form that was based on the World Health Organization for COVID-19. It's a very detailed one. For the hospitalized participant, it takes about 5 hours to prepare. And I'll come back to it. So I've told you about consent. Consent is really important in any studies that includes human participant. And in the case of COVID, it was also a challenging situation because there was a lot of measures that were put in place to prevent contamination. And also we had to, especially early on in the pandemic, limit the use of PPEs, personal protection equipment. So no research personnel were allowed to meet the participant in the hospitalized setting. So it was only the clinical teams that were there. And in order to minimize the burden on the clinical team, what what was done for hospitalized participants, so we had a short verbal consent, which basically was a member of the clinical team asking the person present if they were willing to be contacted by the research team by phone. But because we could contact them by phone, then there was no in-person presence. And the ethics committee agreed that under the circumstances, because we couldn't be allowed to bring papers or pens inside the rooms where patients were in isolation, we couldn't get a signature at that time. So they said the verbal consent will be fine as long as once you've recorded their verbal consent, later on we would send by post our consent forms that the participant fills in and send back to us. So that's the procedures for hospitalized participant. And in the case of non-hospitalized participant, these are ambulatory participants that are coming for our study on long COVID. So when they come, they're no more. It's usually 30 days following the onset of their symptoms, so they should be no more contagious. So in this case, we can do the more classic in-person written consent with the research team, or we also have the option of sending them ahead of time the consent form for them to sign and send back. So you see, the consent procedures have to be adapted to the different scenarios, and this was a very peculiar situation where there was an important need to set up a biobank, but we had to respect the even more important needs of the patients and the treating clinical team in situ. But we were lucky, and everybody collaborated very well, which enabled the procedures to go ahead. So just to give you an idea of some of the challenges we faced, the mandate that I told you about was given by FRQS and Genome Quebec on March 19. The steering committee, the task force that was formed to create the biobank, occurred 2 days later. And we generated the management framework, consent forms, standard operating procedures for bedside and bench side. All of these documents were prepared in the space of 10 days. I can tell you that a lot of people worked very hard and very late in order to get this done, because that would usually take up to a year. And this wouldn't have been possible with the really involvement of, obviously, as I said, many individuals. But I'd like to particularly say that we were in constant conversations on the phone with members of the ethics committee, asking them, what were they expecting, what would they like, what would be feasible in the consent procedures. We set up Zoom meetings with them, so they were really active participants. So I think that was a key to the success, this open communication. We had one goal. We all realized this was going to be important but a challenging situation. So how could we adapt to this? And they would help us. We would send them our management framework, give us feedback. This is okay. This is not okay. And it was a very, very intense period. I can vouch for that. I wasn't trained in all the types of writing that is required in putting these. Luckily, we could use some sections from the management framework from the Quebec Respiratory Health Network Biobank, so that was a good thing to leverage some of what was already there. But there were so many new things that a lot of these had to be rewritten in the space of a little bit less than 10 days, because the project was approved within 10 days, and on April 1st, we could start recruiting our participants to the biobank. So that was a huge success in putting this together very quickly. This is also why, right from the start, we made these documents available to everyone in order for other biobanks that wanted to set up in the context of COVID, that they could just download them and adapt them to their own situations. So let's look a little bit more into the clinical data collection I was telling you about, because as I said, the biosamples are very important, but the clinical data is also tremendously important. Otherwise, you don't know the outcomes of what you're finding from the biosamples, from the molecular study that you're carrying out on these biosamples. So there's a slight difference, I won't go into the detail, between the hospitalized and the non-hospitalized cohort in terms of actual information collected. But generally what we get are the symptoms at the moment, during the acute phase of the disease, presence of comorbidities. Did the person have diabetes, non-cardiovascular disease? We had a fairly long list of comorbidities that were collected. Vital signs were collected, heart rate, pressure, but also blood works. So we collected the types of white blood cells that were in the blood, classical parameters that are creatinine, urea, so things that are done routinely and that are put into the medical dossier. Then some of these information were extracted and put in a de-identified version, de-identified form, where we could have access to this information. We collected medication, some complications that may have occurred during the hospitalization, and a fair amount of just other information such as smoking status, whether the person was admitted to the intensive care unit, if the person required mechanical ventilation. Just to give you an idea, when you open the clinical data file from the biobank, it comes to 900 columns. And currently we have 40,000 lines. So this is not it's a pretty big Excel file. So make sure that your computer is ready to open it. So there's a lot of information. As I said, it takes, for the hospitalized participant, where it is longer, it takes 5 hours on average to just gather all the information for one person. And we have over 4,000 participants that were recruited into the biobank. So it shows the amount of time that it requires. All of this information is then fed back into in our central database, where we generate that Excel file. It's not in Excel, but it can be exported as an Excel file that you can access if you've been granted access, and you can use to make your own studies. So that's the clinical data. The other aspect that is very important are the biosamples that are being collected. Collecting biosamples, you have to be, I remember when we initially thought about what we wanted to collect, we had a huge list. You want to collect as much as possible. So obviously blood, we wanted to probably get nasal swab. We said, some groups were interested in doing the microbiome study, so knowing whether the gut microbiota was affected, so they wanted to get stool samples. Someone said, well, urine would be great. So we had this, big list. Then suddenly you have to go into a more realistic mode of thinking to say what's feasible. Again, we have to remember clinical teams are being challenged by a pandemic. So we cannot ask, because we cannot send a research team in, this has to be done by the nurse that is at the bedside. So collecting blood is not too disruptive. And we couldn't collect blood, and I'll show you the time points. We couldn't collect blood outside of those that were indicated for treatment, so by the doctor. So we couldn't say, if there was no blood that was asked by the doctor, we couldn't collect it at that time. So we focused on the blood, from which we extracted different samples. We extracted the DNA to do genomic studies. We extracted a subset of cell population, cells, leukocytes that are involved in the immune response, because they are certainly very important in reacting to the infection and understanding how the disease may be progressing. We collected the plasma, so when you collect blood, you can spin your tube and separate it either in serum or plasma. There's some slight differences, but basically you get rid of the red blood cell, and this is the liquid that contains a lot of mediators of the inflammatory response, for example, of markers of what's going on in the body that we can use to better understand disease. We collected also another nucleic acid, RNA, to look at the overall expression of RNA in the different cells that were present in our blood samples. And that can help us understand what are the responses to the infection or to the condition of the person that is there. All of these samples were collected at the bedside, processed in labs of different institutions where we had sites that were collecting in the 9 different hospitals, and then they are stored in cryovials, usually in -80 freezers, except for the cells that are kept in liquid nitrogen. And then these accumulates very rapidly. You get thousands and thousands of these tubes. So you have to be able to track those. And I won't talk about it today much, but this is very important. If you don't know, if you collect a tube and let's say you don't write on it what it is, and even then writing is not ideal, the best, depending on the tubes we're collecting, the ideal scenario is that they have now these barcodes that can be scanned and linked to the number that we assign to each participant so that they're de-identified. Otherwise, it's useless. So you have to be able to keep track of all of these thousands of tubes, knowing what they are, when they were taken, and to which participant they are associated. So that's also just a software in itself that is used, that is actually has been developed in Sherbrooke and specifically adapted for the COVID-19 biobank. So if I come back to the different milestones of the biobank, we started recruitment on April 1st. It went really well early on, so we were able to recruit our 1,000th participant in June of that year, and we started releasing data. These were early releases in July of that year. We have now much more data, and so what I'll do now is show you a little bit the state of the collection as it is currently or in the last few months. So the entire cohort has over 4,000 participants. Most of them are COVID positive, but about a little bit less than a third are COVID negative. So they're SARS-CoV-2 negative. As I said, this is really important. And what's interesting in this case, especially if you're looking at the what we call the severe illness cohort, which is the cohort that is hospitalized, these would be persons that came up to the hospital with the suspected infection, symptoms that were likely to be COVID, but they weren't. So they're very good control if you're trying to see not the difference between healthy and COVID, but between COVID and other infection. So that is a strength of the current cohort that is being recruited. We also have this milder or asymptomatic cohort that comes later after their acute phase, but they're followed up over a span of 2 years in order to study the long COVID. If we look a little bit more in details again, it's pretty well balanced in terms of sex. We have more or less half males, half females that are participants, so that enables us to study the impact of sex and gender on outcomes of COVID-19. We don't have a big cohort of pregnant women, but we have 58 in the study. You'll see that more than half have more than 2 comorbidities in the cohort. That's to be expected. The disease affects more the elderly, and the older you are, the more likely you may have a comorbidity, and also you're more likely to be hospitalized if you have comorbidities than if you do not. But we do have participants that do not have any comorbidities in the biobank itself. Out of the 4,000 participants, what's interesting, and I'll show you in the next 2 slides, the design, this is a longitudinal cohort. So we're following, as long as the participant agree and come back, the same person through time. So we have more than one visit. And currently we're now at over 10,000 visits collected in the biobank. And you see the breakdown of those visits. This was up to December, but now we have additional visits, including those coming from the latest Omicron wave. So that's another interesting aspect, is that we started very early, right at the beginning. So we have the first, second, third, fourth, and fifth wave of the pandemic, from which we were able to recruit participants and have samples. And in the future, researchers that want to study the impact of the host response from one wave to the other, they'll be able to do it. So this is the design of the study. As I said, the BQC19 as a biobank is slightly different, as the cohort is built in the biobank itself, which is not always the case. In the case of hospitalized participants, the day 0 is their day of enrollment, and we collect blood sample and clinical data, and that's going to be the case for every single day. And then we do a follow-up on day 2, 7, 14, and 30 if they remain hospitalized that long. So thankfully, and hopefully most of the participants will have left before, let's say, their day 14 visit, but we do have a number of participants to the biobank that were hospitalized for up to 30 days, and we would collect blood and clinical data on those days, or either plus or minus 3 days, as I said, because we couldn't send a research team to collect on a specific day, but we were synchronizing with the indication for the clinical indication for blood being drawn. And naturally, this information is captured in the data that we have in the biobank. So you know whether in theory it's day 2, you know if it's day 1 or day 3. So we know the exact time that is done. That's part of one of those 900 columns that are in our data file. Following hospitalization, we follow them using a same design as we have for a non-hospitalized cohort. So in the case of the non-hospitalized cohort, the recruitment usually occurs at day 30, where we do enrollment and ideally collect blood samples and clinical data. And then we follow at day 90, 6 months, a year, 18 months, and 2 years. This is important because, as you are aware, a number of persons that have had COVID will present long-term symptoms. They tend to vary, and we need to understand what's going on. If you consider the amount of persons infected in Canada with SARS-CoV-2, well, there's a potential for having a lot of people developing one form or another of long COVID. So we need to be able to support the research and the teams that are interested in answering those questions by collecting data and samples that they can use to answer their questions. So that's the overall design of the biobank. So we take samples, data, we put it in a central repository. And normally people would access the samples, do their analysis, and then use the clinical data to answer research questions. So I said there's a bit of a twist in the BQC19, and it's that we did some large-scale experimental analysis of the data that is made available to the community first. So it's not made available to researchers in particular, it's made available to everyone. And these large-scale analysis are quite often referred to omics analysis because they're looking at kind of painting a big picture of what's going on. So we have a list of multi-omics analysis that were done, not on all samples. These types of analysis are extremely expensive, so we have to select a subgroup of participants to the biobank for which we do these analysis. And approximately in this first 2 years of the project, we did this on 2,000, a little bit less than 2,000 visits, which represent 1,200 and some participants. What are these types of analysis? We looked at what's called the proteomics, so the proteins that are found in the plasma, so in the blood, using two different technology, a new technology that doesn't give you absolute measurement, but that can compare one person to the other to say who has more of that protein. We're in the process of doing a more classic quantitative measurements of 48 important molecules, proteins that reflect the state of inflammation in a person, whereas in this sample, we look at 5,000. So you see there's a big difference. So qualitative on 5,000 or quantitative on approximately 50. We have performed genetic analysis, so whole genome sequencing, where we sequence the entire genome of each participant. So that enables to do very detailed genetic analysis. And because the whole genome sequencing is a very big file, we had someone that wanted to download the file on their computer, and they contacted us and said, well, there appears to be a problem. Well, we calculated that with their speed of connection, it was going to take 60 years. So that's not something that you're really willing to wait for. So there's so much information. So there's really big files that are hosted on the Compute Canada server, and you would probably analyze them in situ rather than trying to download them on your laptop at home via your not so speedy connection. The GWAS these are, that stands for Genome-Wide Association. These are SNPs, single nucleotide polymorphism, that are marked and enables you to kind of a shortcut for the whole genome sequencing. So you can look at these marks on the genome in different participants to extract information about the disease progression. And so we have those two types of genetic analysis that are available. Transcriptomic is, instead of looking at the DNA, we're looking at the RNA. And as I said, this is done on the blood and really reflects the RNA content of mostly the cells that are found in there. And the RNA will be changing based on the milieu in which the cells are found. So if there's an infection, obviously certain genes are turned on to help the host fight the infection, to help the host respond to the current conditions. That would be reflected in the transcriptomic data that is being provided. We have two types of immunoserology. What do we mean by that? We're looking at the antibodies. So this is a very important aspect, obviously, in a viral infection. We know how important the vaccination is in protecting against the SARS-CoV-2 infection, but obviously our own body may make antibodies upon infection. So we measured the presence of the neutralizing antibody against SARS-CoV-2 on a subset of the samples, and also the expression of different subtypes of these antibodies against, for example, the spike protein, which is one of the main protein that has been found on SARS-CoV-2 that binds the ACE2 receptor on those cells that permit the virus to enter the cells. So we can look at the presence of these different antibodies in circulation, and that will provide very important information on whether if the person's been infected, what are the levels of circulating antibody, is the immunity, what we call the waning immunity, decreasing, is it being maintained? So this is information that's derived from those type of data. The metabolomic is, you could see it as a kind of a parent of the proteomics. So instead of looking at protein, we're looking at other types of molecules, much broader sets. They can be food-derived molecules. They can be compounds that are metabolized from drugs. There are numerous metabolites in circulation. The technique measures up to 12,000 of these. We have about significant measurements for 1,500 metabolites in our participant. That gives you information on the state of those metabolites, and again can be used to understand what the disease is occurring. So these are all currently available, and additionally some core analysis are being provided. So this is, I've taken a bit of time to express it because this is specific to BQC19. Well, not just BQC19, but this is kind of a unique aspect that is quite important to its function. Now, we've put in place an access process. We've signed up to the Wellcome Trust statement on sharing the research data and findings on COVID-19. So we want to make this available as much as possible, but we must also respect the ethics and legal engagement we have towards our participant. So any groups that want to use the data, they can be from academia, from the pharmaceutical industry, as long as they work for a qualified institution and that they have approval from an ethics committee for the use of those data to answer a research question in COVID-19. They make an application, depending if it's for data only or for data and sample. We have two pipelines of evaluation. We try to make this as speedy as possible, especially for the data access, the access to data only, because this is a renewable resources. The data we send the data. We're not. There's no chance of not having the data anymore. We've got good redundancy in the servers, but the biosamples, there's a limit to their numbers, so this is a little bit more stringent, evaluated a little bit more stringently by the access committee to make sure that the samples are going to be used appropriately. So if I sum up the uniqueness of the biobank, it's a case control design, as I've shown you. We've got extensive clinical data, longitudinal follow-up, recruitment through all the pandemic waves, in-depth multi-omic score analysis, and an open access policy to favor as much research as possible to be done on COVID-19. When you're looking at biobanking during a pandemic, what were the key features? We had to adapt the consent procedure to infection control. We had to leverage existing resources to enable this quick turnaround in 10 days of getting most of the paperwork done. And obviously that couldn't have been done without teamwork. I'll skip this slide. And talking of teamwork, this is a list. There was over 300 persons that contributed to the creation of the biobank. And I've listed them all here, and I think without them, this would not have been feasible. So this illustrates what can be accomplished when we're working as a group. I'll very quickly give you an illustration of what can be done with the data, and then we'll take questions. So what I mean by post-series science approach is sometimes we make an hypothesis, but sometimes you want to let the data talk first. So the post-series approach is leveraging artificial intelligence quite often and machine learning algorithms to help you see patterns that you may have missed by going with a hypothesis-driven approach. And now, one classic example of this is what we could call the many-one limitation. So if you considered severity of COVID-19, you could say everyone that is severe we put in one group, and we compare this to another group that doesn't have severe disease. But then you're assuming that all the person in your severe groups are there because for the same reason. But that's rarely the case. Usually you have like many roads that leads to Rome. There's many roads that can lead to severity. And in order to capture that, you can look at trying to break your, if you have large data set, you can try to see if some of the participants in your group are more similar to each other than others, and then look at the outcome of each of those. And that's what we did using the data from BQC19. We studied 731 hospitalized SARS-CoV-2 positive BQC19 participants, and we've looked at this proteomics I was telling you about, the 5,000 proteins in circulation. We basically use a computer algorithm that compared the 700 participants, look at each of the 5,000 molecules and classifying them as to which one are closer to each other. So I'm glad I didn't do this, and we asked the computer algorithm to do this 1,000 times just to make sure that the data was solid. And that gives you this kind of heat map that we see where these are the 5,000 measurements on this axis and the 700 participants. And it classified them in different groups. We have 6 groups here, and I won't go into the details of telling you what each group does, but the question was, do these groups have different clinical trajectories? Is our hypothesis correct that some of these groups may have different disease outcome? And that's what we see here. We have a group. This is the time to admission to intensive care unit or death, and we identified a group, the group 6, that is doing much worse than the group 1 and 2. And we probably have an intermediate groups there. And so we were able to discover these groups of patients by simply looking at the molecular data, not the clinical data. And that enables us to probably understand better how the disease progress. So I'll finish here. Just want to acknowledge this is done in a group. So I want to acknowledge my colleagues. The bioinformatics was done by the group of Aminaei, Antoine Soulet and William Ma are trainees in this lab. Karim Tremblay in Chicoutimi, she's worked with us in answering these questions. People at my lab at McGill. These are the funding agencies for the second part that I've told you in 5 minutes. I'll bring back this slide. The Biobank itself was funded by the FRQS, Genome Québec, and the Public Health Agency of Canada. So thank you, and happy to take questions.
Absolutely. Thank you so much, Professor Rousseau, and for such a great and informational presentation. I think this is the first time that I've personally heard of a biobank so in depth, and hearing your detailed showcase about how it has come to fruition and what it involves is extremely interesting, both for me and I'm sure for everyone else here. We'd like to now transition, as you mentioned, into the Q&A session for the seminar, and I'd like to invite members to post their questions in the chat. I believe there's already one in the chat from Samara, and I can just go ahead and start with that. So Samara mentions that we learn a lot about bioethics in school, and she's asking what are, if any, the measures in place to protect the privacy of participants.
Okay, so that's a crucial element. In the biobank, all the identifying information is kept separate from the central database. So there's the identifying data is linked to a number, and we only get the number. So we don't know who is what. Obviously, once you have whole genome sequencing, in a sense, you have identifying information. That's why we have to, each project that will access the data has to be vetted by an ethics committee, a local one, and by the access committee, just to make sure that all these safeguards are in place, that no one will be distributing that information widely, but it will be used only for research purposes. So there's 2 steps. There's one where we separate identifying information from the rest. And the other step is making sure that qualified individuals, and we have questions in our access form that addresses these points, and everybody signs that they will not distribute this freely, that they themselves are engaged legally in protecting that information, making sure you don't let the clinical file lay on your desktop on an unlocked computer in a cafe, for example.
For sure, that sounds very insightful, and it seems like all the safety protocols are in place. So moving forward, another question that was asked by Emma Coster is there any specific findings that have come out of the biobank that you think has been the most significant or impactful finding?
That's a very good and tough question, because the impactful data that is generated will know in the long term. One of the big challenge with a biobank is, as you've seen, it takes time. It takes time to get the data. It takes time for science. If you're trying to see what's going on between different waves, we have to wait until we gather the information. So I think it's really early to say what's going on. I'm really happy to see that our data was used in big genetic studies that have identified genetic markers linked with severity. So this is mostly the type of research that has been done at this stage with the data. And so we list these papers on our website. So for those that are interested in seeing the science, but that's these were the first step. I'm hoping to see more and more. And I kind of didn't answer your question directly, because it's really difficult and it's impossible to know at this stage what will have most impact in the future.
No, absolutely. That makes a lot of sense. It is very, very early on, and I can understand, and I'm sure Emma can as well. But there's another question from Gino. I'm sorry if I am mispronouncing that, but regarding data collection, did you use an out-of-box LIMS or created your own database? Also, did you extract data from patient charts manually or use some sort of AI?
Okay. The data extraction is a mix. I'll start with the second part. We have some of the larger hospitals that were able to do automated data extraction, which saved a lot of energy from individual, but does demand a little bit more work into making sure that the fields have been mapped correctly. And we've had a few errors there, so that it wasn't as simple as we would have wished for. And some other centers weren't equipped, so it's all manual capture of the data, which includes also risk sometimes of mistranscriptions. For the first part, we used the LIMS system that had been developed for the Quebec Respiratory Health Network Biobank, which had its own set of modules that could be modulated. I must say, because it's an in-house developed system, it was really useful in making it specific for the purpose, but it was a little bit more challenging in adapting it to the volume of samples being inputted into the system, because this was it's a biobank that was on steroids, literally growing very, very quickly. And so the LIMS system is a very challenging aspect, and it's working, but you know, we could see improvements, and we're looking into improving it over the coming years.
Absolutely. No, that it seems like you already have it all clear and clear cut. But in the future, I can imagine that it will continue to build and improve. Kunwar is asking, I'm sure a lot of people request to use data for their projects. How do you decide which project gets to use it, and what is the criteria?
I'm hoping there was even more people that would demand access. One of my mission is to get as many people as interested. For data access, the criteria are pretty wide. They're really limited by our consent form. So, you know, our duty is to the participant. And in our consent form, one of the things that is mentioned is their sample will be used for COVID-19 research. So if you're wanting to access, and this is for COVID-19 research, this is the main criteria. If it's for data only, that shouldn't, you know, as long as you have ethics approval, then you will get access. We're not doing an in-depth scientific revision. Usually projects are already approved to get ethics, so they have to have been reviewed by another group. It's slightly different for the biological material because, as I said, we don't want to give out all the samples without really thinking about it and have, let's say, 20 groups request a tube of serum to do the same measurements. We'd rather have one group, and something I didn't mention, every group that do analysis on it, because we've signed for this welcome trust statements, must send their analytical data back so it can be shared. That's where we are acting a little bit more stringently in the access for the biological material. But we're getting so much material that I'm really encouraging as many groups as possible to apply and to really generate experimental data that we can put back in the biobank and then distribute to the entire community.
Absolutely. I think, as you mentioned, it is very, very valuable information. You're getting so much that it's important that like as many groups as possible can use it. And just moving on, I think Maral says, I think Dr. Ask has his hand up. Feel free to speak.
Thank you very much. That bypassed the question, so that if others have questions, please put your hand up and we can unmute you. So my question to Simon was: are you aware of any other biobanks in like this that is set up in a better way, or is this the state of the art as good as art? Do you have any regrets somewhere where you're saying, oh, we should have this element into this biobank, because it all sounds extremely well set up?
You know, there's always regrets, some that we're hoping to be able to countermand. I think some are impossible. You want to make this resource available as quickly as possible, but you have to respect ethics. You have to respect all the legal procedures around it. But setting up what we call the interinstitutional agreement, which is another document I didn't talk about, but the agreement between the different institutions that enable the transfer of the data, took months, and that I wish would have been optimized earlier, and that would require input. So there's an optimization at the level of the paperwork. We need to link with other databases. We're in the process, but that hasn't happened yet, with all the viruses that were sequenced from the different participants. So we have about 700 of those, but it would be really important and super exciting to be able to link the viral genome to the host genome and the host response. And so that hasn't worked out as well as we would have wished. And integration also with administrative data held by the government agency is an absolute nightmare at this stage for us. And I think for many people, I don't know what's the regulation in Ontario and in other provinces, but this prevents us from accessing important data for the research community. So that's also things we would like to see improve. These are some of the key elements I see. We would always like to have more samples, but at the same time, having samples for samples, there has to be a need. It has to be coming from the community, those requests. Some people wanted to have these nasal swabs, but there was no way we could get our hands on those swabs because they were all used for testing. So there were limits that were outside of our capacities.
And the other question was, are you aware of any other biobanks on COVID-19 around the world that has better?
I didn't do. We've tasked a student to do a review of the different biobanks. So I'm hoping that in a few months we'll be able to give you a clearer answer. I think there are biobanks that have, there's a trade-off. There are biobanks that have a lot more samples, but they're not as deeply phenotyped. Some are much smaller data set, but have imaging data, which can be super exciting. So I think every biobank has their specificity. What's important is that we can link to each other and enable researchers to find the resources they need to answer their questions. So yes, there are certainly biobanks that are better in one way or another, but we also are well set up in other ways. So I think it's really defining what we have and what we're missing and trying to build networks and links. And CoVarNet in Canada is doing exactly that, putting together the different biobanks that were created across the country on COVID-19 so that researchers can go in one place and see what's available and build their studies using this.
Very good, thank you.
Thank you for those questions as well. And I imagine that with a data bank or biobank of this size, there are challenges and benefits on both sides, right? So, and just moving on, I believe we have a question. We have Gino's hand is up if he wants to extend his question from earlier. Go ahead.
I think you just need to unmute yourself, Gino. Or perhaps that doesn't work.
Yeah, it's possible it doesn't work. Or you can just post your question in the chat, and I can just read it out as earlier.
Oh, I think so. I have unmuted you, Gino. So you just need to unmute yourself. Your microphone is enabled, but if it doesn't work, then we will need to go in the chat. So I think that Gino's question. Are you there?
Okay. I think Gino has passed. Okay. So I'm just going to move on to some of the other questions that we had. That's quite a bit. Anusha has asked, for hospitalized patients who passed away due to COVID-19 before the day 7 collection visit, would the samples collected at earlier visits, such as day 0 or day 2, still be used and included in the biobank? Or would only individuals from whom all samples were collected from day 0 to at least day 7 be included?
No, we keep all those samples. You'll get in the data file, you'll see when time of death has been put, but you may, for a number of participants, we may only have the day 0 visit. If you're doing genomic studies, that's not so much of a problem as long as you have the outcome in the clinical file. Obviously, if you're trying to look at the longitudinal measurements of an inflammatory marker of a cytokine in circulation, that may be more frustrating. There's many conditions which may make these different samples not available, but because of the it's not tremendously large numbers, but because of the good numbers, we do have a few complete longitudinal sets.
Absolutely. I know that I believe that answers her question, but there's a few more. So Nadia asked, is there any strategies you use to encourage people to continue with the collections over the long period, especially since the non-hospitalized cohort will be enrolled for a longer period?
Yeah, that's interestingly, we have more problem right now with our hospitalized cohort follow-ups. That may be because it's a more elderly population and they went at the hospital and they may not be as interested in keeping on, whereas those that are outpatient are coming, a number of them have long COVID, so they're willing to come longer. But to really answer your question, we have actually a team that is set up by my collaborator, Karim, they're doing exactly a study to identify the barriers or why people are dropping out of the study. They just completed 23 interviews of different participants, and I know they're collecting the data right now to do some of these analysis so that we can adjust our strategies in order to ensure that we keep our participants as long as possible, because there's a big value in longitudinal sampling.
Yeah, I can imagine. I think that's a great answer. And just moving on, Smeet is asking, regarding the biobank, would one be limited to only running correlational studies using the biobank, or would there be possibilities to run other types of studies?
There may be, right at the top of my head, I cannot go through the entire set of potential studies, but there is, because you're not doing an intervention, it's hard to see. There may be interventions that have been done, and therefore you can leverage that information. So if you're interested, for example, in the impact of corticosteroids, you may be able to extract sufficient participants for which they were given a corticosteroid. But obviously, this wouldn't be done as neatly as it would be in let's say, in a true interventional study. So yes, there are other possibilities, and one could also take cells from, let's say, take PBMCs, and some groups, they tell me they've had some successes at trying to challenge them. I don't know if they were kept in the conditions that were okay for that. But yeah, it's not just correlation studies. There's other possibilities. But yeah, because the design was made to be as wide as possible, obviously it ironically limits some of the potential for certain types of other approaches or studies or designs.
Absolutely. And just moving on to one other question we have is, Nadia also asks, as we all know, COVID is a rapidly changing predicament. So how did it feel at the beginning of the pandemic? Was it overwhelming? What kind of impacts did the emerging variants have on your work?
Yeah, it's funny. It was very, at the beginning, the task was immense, and we were, the steering committee was made of 10, 12 researchers. We were meeting 2 hours every day, 7 days a week. And that's just the times we were meeting. And after that, we had to, work on the documents and all of these things. So that was a pretty big impact. And that lasted for at least, I think it was at least a month, if not 6 weeks of very, very intense work to get all of this going. But then you look around and you see what's going on, and a lot of things are stopped, and you feel, engaged in this task because of the importance, the impact it has around you. when we started seeing the variants of concern emerging, then again, you see it kind of reinforces the importance of keeping this ongoing. When Omicron, our initial budget was for 2 years, which officially finished in theory last week, yesterday, sorry, March 20, but it's to the end of this month, but we've had an extension. But when Omicron hit, we didn't have sufficient funding to capture that wave because we didn't expect to have such a big wave. So we had to request for emergency funding. So that demanded, again, with every wave, as hospitalizations go up, our activities in the biobank follow accordingly so that we can provide the samples to the community. But it's not as bad as it was early on.
Yeah, no, I can imagine at each step, there's like a little bit of a challenge and a hassle you have to go through, especially as you mentioned with funding. And that kind of leads into Maral's question, which is how much having more community involvement in the data collection step can affect the biobank and its analysis.
Yeah, it's really important. For me, one thing that I found truly attractive about this project, it's a community project in a sense. It's the research community working together, trying to generate this as a group. And in our academic careers, we're quite often, we're working together as a team, but we're also, trying to achieve and competing a lot with each other. But I think this is a case where the working together, the community aspect came out further. So if we can really engage, again, the community to keep on accessing the data, doing analysis, generating new data that's fed back, we have to realize that this is a unique opportunity. Not many diseases will be as studied as COVID is. And that may benefit other syndromes that lack the depth that we're currently having. So I think we truly have a unique opportunity to study something in a way that we couldn't have done for not many other diseases on the planet, and that we have to leverage this, just kind of see what we can do when we put all that amount of energy worldwide. I'm not just talking about our own biobank, but the entire, the publication number is crazy. The amount of work that's been done on COVID-19 is crazy. And let's see what we can learn from all of this. And there's so much that we cannot do it alone. We have to work together.
No, I imagine, I think this has been a theme for the entire pandemic, pretty much. No one can do this alone. It's always been a group issue. It's affecting us all, and we have to work on it together. But we do really, really appreciate your time. I can tell that we were just nearing the end of the allocated time, and we do want to let you know that we really, really appreciate you being here for the last almost 90 minutes. And your time is valuable. So we really appreciate it. I just want to ask the students of Health Science for DM3 to please fill out the feedback form. I believe it's in the chat. Dr. Ask has just posted it. So we'd really appreciate if you went in and filled out that form. And there was just one question from Gino. He was wondering about he himself is working on he's in the early phases of exploring the feasibility of creating a multi-institutional biobank, and he was wondering if you can connect with you outside this presentation. So I'm not sure if that's something that's feasible for you, but
but I'll be. You can connect with me by email, and I'll be happy to give him the help that I can get. I think there are challenges specific to multi-institutional biobanks, but it's really worth the effort.
Absolutely. Okay, so Gino, you have your answer. We really do appreciate your time, Professor Rousseau, and thank you again so much for coming.
I want to thank you. It's all our time is precious, and not just mine. So I'm happy that you took the time to be there, and it was great feedback and great questions. So hope I was able to shed a bit of light on biobanking.
Thank you very much. Thank you all.